It's a pleasure to be here to present our work on adolescent development of functional brain networks. The reason we're interested in adolescence is because numerous psychiatric disorders emerge in this period, but the literature on functional network development in this period is not entirely clear. For example, many early studies reported on a distance-dependent development of functional networks with a preferential weakening of short connections and strengthening of long connections. However, it appears that this was possibly driven by in-scanner motion of the subjects, with younger children tending to move more than older adults, and with motion having a differential impact on short and long-range correlations. Another possible confound is the relationship between many higher-order graph theoretical measures calculated on fixed density networks, that is, networks where a fixed proportion of the strongest correlations is retained in each subject, and properties of the underlying functional correlation distribution. And finally, there are fewer studies on subcortical connectivity, or the studies that have looked at this have generally looked at specific subcortical seeds. So our aim here was to revisit the functional development literature, first of all using a large new data set of around about 300 adolescents aged 14 to 24. This is data from the Neuroscience and Psychiatry Network, which is a large collaboration between the University of Cambridge and University College London. These subjects were scanned in the resting state using multi-echo acquisitions, which allowed us to apply multi-echo ICA, or MICA, which is a principled method for removal of noise or of non-bold components based on dependence of the bold signal on echo time. Second, due to the aforementioned relationship between higher-order graph measures and the underlying correlation distribution, we focused on unthresholded or fully weighted networks, which we extracted using Pearson's correlation between 333 regions of interest, including 16 subcortical regions from FreeSurfer and cortical regions from the Glasser parcellation after excluding dropout regions. And finally, we focused at the region level both at cortico-cortical as well as cortico-subcortical maturation, including individual subcortical regions. We modeled maturation as linear. We also fitted nonlinear smoothing splines, but they did not provide evidence of a better fit than the simple linear models in these data. So looking first at the functional correlation distribution across all subjects with here age color-coded along the y-axis. We summarized changes in the functional correlation distribution using the 4 moments, and we found an increase in the mean and in the variance of these distributions. So these distributions are shifting rightwards and expanding, and a decrease in the skewness and kurtosis, which essentially means they're getting a peak here and the tails are shifting outwards. And you can hopefully appreciate some of these changes if I show you averaged distributions within 5 age bins, which are then overlapped. And so in the top inset, you can see a leftward shift in the peak of the distribution and an outward shift in the tail in the bottom inset. We next looked at maturation in these distributions in greater detail by fitting linear models as a function of age to the weight or correlation at each percentile of the distribution. Here you see the same distributions now color-coded by percentile. And on the left, you see linear models as a function of age fitted to these percentiles, and on the right you see the slopes of these linear models. And hopefully you can see that as you move towards the right tail of the distribution, the slope or the rate of increase in connectivity strength becomes greater. And you can appreciate that if I show you the zero-centered slopes, where again, as you move to the right, the rate of increase becomes greater. So the strongest correlation strengthens fastest. We next looked at regional maturation using functional connectivity strength, or the average connectivity of a region to other regions. We calculated 2 flavors of this. First of all, cortico-cortical node strength, or the average functional connectivity of a region to other cortical regions, as well as cortico-subcortical node strength, or the average connectivity of a cortical region to subcortical regions. And we fitted linear models to these 2 versions of node strength as a function of age, and we extracted 2 parameters from these linear models. On one hand, the intercept of the linear model, which is indicative of connectivity strength at age 14, And this shows similar profiles in both corticocortical and cortico-subcortical strength, with primary cortical areas showing the strongest connectivity at age 14. Second parameter we extracted was the slope of the linear model, indicative of the rate of maturation over adolescence. And this shows in corticocortical strength the greatest rates of increase in primary cortical areas, but in cortico-subcortical connectivity, this differs and shows greatest increases in strength predominantly in association cortical regions. And you can appreciate the difference between these two flavors of node strength by calculating the relationship between the maps at baseline at age 14 and the changes as a function of age. And so for corticocortical strength, the relationship is positive, with regions that are more strongly connected at baseline showing a higher increase in node strength. In other words, the richly connected regions get richer. And in cortico-subcortical connectivity, this relationship is negative, with the regions that show higher connectivity at baseline showing a slower rate of increase or even a decrease in node strength over adolescence. In other words, the poor get richer or the rich get poorer. Knowing that different subcortical regions show distinct trajectories of maturation, we repeated this analysis for connectivity between individual subcortical areas and cortex. And here you see that the individual subcortical areas show different profiles at age 14, but you can kind of see hopefully that generally the sensory motor areas show stronger connectivity at 14, and the association regions show weaker connectivity at 14. And looking then at the changes as a function of age in this connectivity, again, there are differences between the individual subcortical regions, but generally the association regions that are weakly connected at 14 tend to increase in their connectivity to subcortex, while the primary sensory motor areas that are strongly connected at 14 tend to decrease. Finally, we looked at maturation at the level of individual edges or individual correlations in the matrices, and we fitted linear models as a function of age again to each edge. And again, we extracted functional connectivity at age 14, as well as the slope as a function of age at each correlation. And we are interested, again, in this relationship of baseline architecture to changes as a function of age, but this time at the level of individual regions. So asking, how does the profile of connectivity of a region to other regions at 14 relate to its change over age, over both cortical and subcortical regions? And so to look at this, we correlated rows of these two matrices, and we found the following pattern with strong positive correlations in primary sensory motor and visual areas, and negative correlations in association cortex, and below you see subcortical areas. So the strong positive correlations in primary areas essentially again are indicating that the greater the connectivity at baseline of a primary region, the greater its rate of increase. So the rich get richer, but the pattern of connectivity is stable in its topology, while the association and subcortical areas that show greater connectivity at baseline show a higher rate of decrease, and vice versa. So there's more of a qualitative reorganization of connectivity in these regions, with the rich getting poorer and the poor getting richer. So some of the work we're interested in pursuing further is looking at the relationship of these patterns to maturation of structure, which we've previously reported on in these data, including cortical thinning and intracortical myelination, predominantly in association cortex. The relationship of this to structural networks, again in these data, which again show changes in structural correlations in association cortical areas that are related to the changes in structure. So it'll be interesting to study the relationship of all these patterns to the changes in functional connectivity that I've just shown you. We'll be extending this to longitudinal data, so we have follow-up scans on most of these subjects. And ultimately, we're interested in the relationship of this to cognition and to disease, which is the main reason why we're interested in adolescent maturation in the first place. So to summarize, I've shown you that the functional correlation distribution presents a non-uniform shift over adolescence. And this might have driven some of the early graph theoretical studies on adolescent maturation. And regionally, the primary cortical areas that are strongly connected at baseline strengthen, but their functional connectivity patterns are stable in their topology, while association cortical and subcortical connectivity undergoes more of a reorganization or rewiring. So this is work done together with a large number of people, in particular Ed Bullmore and Petra Vertes in Cambridge, as well as many other collaborators in Cambridge and at UCL. I'd like to thank the funders for their support, and thank you for your attention.
Thank you. We have